yesterday we kind of started talking, we spent some time talking about some of the advantages and disadvantages to asexual and sexual reproduction. And we talked about how if we're going to do sexual reproduction, we can't do mitosis, we have to do meiosis, we have to do a reductive cell division so that the egg and sperm each have half of the amount. And so when we combine them, we're back to having, right? And so we introduced some terms, diploid, which means 2 copies of every chromosome, and haploid, which means you just have 1 copy of each chromosome. And so in this first part here, you're just supposed to identify whether it's describing a diploid or haploid cell. So if you're not familiar with the word somatic, soma means body. So these are basically most of the cells in your body are somatic cells. And so in that case, most of the cells in your body are diploid. Okay, yeah, you have all 46 chromosomes, 23 for mom, 23 for dad. Gametes, that's another word for reproductive cells, for egg and sperm. Those cells are going to be haploid. 1N stands for haploid, 1 copy. 2N stands for diploid, 2 copies. If you have homologous chromosomes, then the cell is going to be diploid, because homologous means they're the 2 of the same, right? So you have 2 of each, it's diploid. If you have 2 sets of chromosomes, diploid. If you have 1 of each chromosome, again, that's haploid, right? So definitely need to know those 2 terms and some of the different cells they're associated with, what it means. This part here is really just some math. So if a diploid cell has 8 chromosomes, then a haploid version of that cell is only going to have 4. You can think about it, if this was 2 in math, 2x equals 8, then 1x would equal 4, right? So you can think about it kind of mathematically too. If a haploid cell has 12 chromosomes in an organism, then the diploid cell of that organism would have 24, right? Some organisms, especially plants, actually have more than 2 copies, so they're going to, in some cases, be tetraploid. The prefix tetra means, well, tri means 3, tetra means 4. So if we have a plant that's tetraploid, it has 4 copies of each, in total 12 chromosomes. Then if you had a haploid cell, it would have 3. And if a diploid cell for an organism had 16, then a tetraploid organism would have 3. Okay, so we'll use those notations a bit more today, just kind of give you a little more practice with that. And then I think this is a good, really important question. If we have a cell with 20 chromosomes and it goes through mitosis, each of the resulting cells are going to have 20 chromosomes, right? Mitosis, you're making identical cells. If that same cell with 20 chromosomes goes through meiosis, the resulting cells are only going to have 10. Okay, so we're reducing it in half, right? So meiosis, if the parent cell has 20, our daughter cells, our reproductive cells that we make are going to have half the amount, are going to have 10. So yeah, I think that's all pretty important. Any questions on that before we move on today? Right, yeah. Not much on the board yet today. Okay, so we're going to take a look at the stages of meiosis today, and that's why I gave you those sheets to kind of help fill in so you don't have to draw all the circles. You'll notice that on one paper it says meiosis 1, and on the other paper it says meiosis 2. So the process of meiosis actually has 2 rounds of cell division. So we're going to start with meiosis 1, and then we'll move on to meiosis 2. It's a pretty long process. Okay, so we're basically going through cell division twice. So before we actually get into the stages, just want to point out again the life cycle for organisms that reproduce sexually. We spend some part of the life cycle as haploid cells, as gametes, and the other part of the life cycle is diploid cells. So here's our original gametes. Here's our egg from mom, and here's our sperm from dad. You'll notice they're haploid. They have one of each chromosome. And so when we go through fertilization and we combine the genetic material in those two cells, now we have a diploid cell. It has two copies of every chromosome. So now we have homologous chromosomes, right? This longer pair is homologous, and then it's a little bit harder to tell size-wise which one's paired up. But we have 3 pairs of homologous chromosomes now. We go through meiosis again. Those chromosomes are going to get mixed up. So in this case, this must be the male. So when he produces his sperm, those chromosomes that he originally got from mom and dad are going to get mixed up in his reproductive cell. Okay, so this process of meiosis, not only are we reducing the amount of genetic material in half, but we're also doing a lot of mixing here. So it's kind of, you know that you have half of your mom's DNA and half of your dad's DNA, but you could potentially have, 60% of your maternal grandmother and 40% of your mother's material. I'm not making any sense here. But the amounts of material you're getting from your grandmother and grandfather could potentially be different from each other, because you can see here they have different amounts of his paternal and maternal chromosomes going in here. So not only are we doing a reductive process so we can combine, but we're also mixing genes up as we go along. So we're not going to start drawing yet, but this is just showing you the whole process of meiosis. So take a couple seconds to walk through this with your group members. So what do you think is happening in each one of these stages? So it starts here, and then it goes around this way. What do you think happens? Just talk to your members.
Oh, yeah.
Oh, I'm sorry. It goes from here and then it restarts here. My bad. That would be really confusing.
Yeah.
So we mentioned there's 2 rounds of division here. So this first row of pictures, this is meiosis 1. So we have interphase. We have prophase, where our chromosomes condense and our nuclear membrane breaks down and our spindle fibers start to form. You'll also notice this doesn't happen in mitosis, but our homologous chromosomes have paired up with each other. And there's a process we'll talk a little bit more about here, but they're actually, the sister chromatids are actually swapping sections of DNA with each other. And then we move on to metaphase 1. Notice the chromosomes are still paired up with each other. We move on to anaphase 1, and the homologous chromosomes are separating from each other. So this is similar but different to mitosis, right? So the homologous chromosomes are separating from each other here. And so as a result, we end up with these two cells. So that's the first round of cell division. Really important thing to notice here is that we're starting, look at our notation here, 2N, which means that we have 2 copies of each. It's diploid for a total with a total of 4 chromosomes. When we get to this point, now each cell only has 1 copy of each. It's haploid and only has 2 chromosomes. Okay, so the reductive meiosis 1 is the reductive division where we're going from having 2 copies of each chromosome, 4 in this case 4 total, having 1 copy of each homologous chromosome. And then each one of those cells is going to divide again. So these are going together. So this is prophase 2, and then metaphase 2 when they line up, and then anaphase 2 where the sister chromatids separate, and then telophase 2. So we end up with a total of 4 cells, and each one of those cells is haploid and has 2 chromosomes. So again, we're going to go through this entire process again, but this is just kind of an overview of it. Questions so far? Yeah.
So I'm confused. Go from anaphase 1 to, or telophase 1 to telophase 2?
Yeah.
So wouldn't it be making 4 cells in total?
Yeah, that's what I said.
Oh, okay, well,
At the end of the first division, there's 2 cells, and then at the end of the second division, then there's going to be 4.
so the at interphase 1, where there's 2 of those yellow things,
Yeah.
those that gets split in half at the very end, right?
Yeah. So all this massive DNA are these chromosomes. So in this particular, we have way more than this, but in this particular model, there's two homologous pairs, 4 total. And so in the first round of division, we're separating the homologous chromosomes from each other, and that gives us half of the amount of chromosomes that we started with. Yeah. And then we go through the second round and we pull apart the sister chromatids, and then we end up with 4 cells total. Yeah. Yep. Yeah, so end result, we end up with 4 cells. Each one has half of the amount of chromosomes as the parent cell. There are some differences in the way that we make sperm and the way that we make eggs that we'll talk about tomorrow, but in general, that's the idea of meiosis. Okay. So you're going to want to do some drawing here probably on your sheet. So we're just going to color out these chromosomes. You don't have to have red and blue, but what do you think these red and blue are supposed to represent? Well, yeah, the chromosomes. So why are they different colors? Yeah, so the red ones can be the maternal and the blue ones can be the paternal. Whatever colors you have, one color is the maternal chromosomes and one color is the paternal chromosomes. And so in that first cell, we're going to start with a diploid cell with 4 total chromosomes. So we have 2 longer chromosomes and 2 shorter chromosomes, one from mom, one from dad. So 2 homologous pairs. This would be G1 of interphase. And how do we know it's G1 of interphase? They haven't replicated yet, right? So they're still individual chromosomes. They haven't gone through replication. And so again, our notation is going to be 2n equals 4. Again, because we have 2 copies of each, it's diploid in a total of 4 chromosomes. Since they haven't replicated yet, these chromosomes are all single-stranded, right? So we're starting off with a parent cell in G1, 2n equals 4, single-stranded. And like we mentioned in the overview, this first division of meiosis is going to separate homologous chromosomes from each other. So we're going to go from having 2 copies of each chromosome to having only 1 copy of each chromosome. So by the time we get to prophase 1, our chromosomes have replicated. So now they're no longer single-stranded, they're now double-stranded. So our notation is still 2n equals 4 because we still have 2 of each chromosome. It's just now each one of those chromosomes is duplicated, double-stranded. And something you hopefully noticed, this does not happen in mitosis, but these homologous chromosomes are pairing together. So that pairing of homologous chromosomes only happens in meiosis. It doesn't happen in mitosis. We have a special name for that pairing of chromosomes. So the process of this pairing is called synapsis. So usually synapse means some kind of joining. And these chromosomes together, when they're paired like this, are called a tetrad. And so that comes from the fact that tetra means 4. And so we have 4 sister chromatids that are in this structure. So you can say the homologous chromosomes go through synapsis. You could say the homologous chromosomes form a tetrad. You don't have to redraw that. You can just point to it yourself. There's a really special event that happens in the formation of the synapsis and in the formation of those tetrads that you might remember from Bio 1. And that process involves the trading of pieces of DNA. The technical term for that process is called crossing over. So this happens during prophase 1. It's called crossing over because the sister chromatids like literally cross over and intertwine with each other. So when they form that tetrad, they overlap. This is in part because, remember, they're identical pieces of DNA, so they have the same genetic code, and so that's going to allow the base pairs to match up with each other, kind of overlap. And in that process of intertwining, they'll often swap pieces. So I'm sorry this is really low down to the screen. Well, maybe I'll move it up. I want to add some genes onto these chromosomes so you can look at. So we have something similar to this. So we're going to look at a gene here. Let's just say it's gene A. And so on my maternal chromosome, I have a dominant copy, and on my paternal, I have a recessive copy of that gene. And we'll we'll put gene B down here. And just to make it easy, mom will have both dominant copies and dad will have both recessive copies. So when they swap DNA, each one of these sister chromatids is now, well, this kind of gets messed up because the same pieces are getting swapped. This is a terrible example. But you could potentially see how when the genes get swapped, you could end up with different sister chromatids. I'm gonna no, that doesn't make any difference. I need to change this. Why? But what's basically happening, when you swap these alleles, you could end up with potentially 4 different sister chromatids, and that's a horrible example of it because they crossed over on both of the genes. But that process is going to mix up the versions of genes that are on each sister chromatid. So this diagram shows it a little bit better. That process of crossing over has 3 steps. So first of all, the sister chromatids have to literally overlap each other and cross over. There's a process where they both break right at the same location. And then they refuse, but on the opposite sister chromatid. And so that's going to end up with a new combination of traits. So sorry, this will be better what I was trying to demonstrate. So before crossing over, here's the paternal chromosomes. They all have the exact same sequence because they're copies of each other. Here's the maternal sister chromatids. They have the exact same sequence because they're copies of each other. When they cross over, only certain parts of them are going to end up swapping. So now this sister chromatid has all of the dominant copies of the gene. This sister chromatid has the dominant A, but recessive B and C. This sister chromatid has recessive A, but dominant B and dominant C. And this has all recessive. So as a result of crossing over, now each one of these sister chromatids is unique from each other. Okay, they all have different versions of the gene. So that's what I was trying to show you on that last slide. Sorry about that. That was terrible. So again, the advantage of this is we're making new combinations of genes. And from yesterday, this mixing of genes is especially helpful when you have populations that are in stressful environments, right? And this actually helps to mix up some of those harmful mutations. So we're not passing if harmful mutations pop up, we're not passing them on to every single one of our offspring. So after prophase 1 is going to come metaphase 1. Just like metaphase and mitosis, the chromosomes are going to line up along the middle of the cell. Different from mitosis is that they're going to stay paired up, right? So instead of having sister chromatids on either side of the plate, we're going to have homologous chromosomes on either side of that metaphase plate. And we're actually going to skip drawing anaphase, but what's going to happen in anaphase? They're going to pull apart. So one homologous chromosome is going to go to one side, the other homologous chromosome is going to go to the other side. And so the end result and again, sorry, now it's getting really low to the bottom of the screen. If we skip anaphase and draw telophase 1, Now, each one of those cells has one of each homologous chromosome. I tried to indicate that crossing over happened. So at this point, we now have different genetic material in either one of those cells. Notice at this point, our notation for the cells has changed. So we're no longer diploid. We don't have 2 copies of every chromosome. We only have 1 copy of each chromosome for a total of 2 chromosomes. And the chromosomes are still double-stranded. So we haven't done anything. We haven't separated our sister chromatids yet. I'll let you finish drawing that out. Any questions on the first round of division on the meiosis one? Meiosis 2 is going to look a lot more like mitosis, just with half of the number of chromosomes. So in meiosis 1 here, we divided the homologous chromosomes from each other. That's the reductive process. In meiosis 2, we're just going to end up splitting the sister chromatids from each other. So if you want to switch over to the second sheet I handed you, it has the circles drawn out already. How helpful that really is. So we call the second round of cell division meiosis 2. And like I mentioned already, in the second division, we're going to separate out the sister chromatids from each other. So we're starting with the same cells we ended with in meiosis 1. We now have 2 cells that are in prophase. So depending on what type of, if we're making egg or sperm, the timing difference between meiosis 1 and meiosis 2 is different. So you might have cells in telophase 2 in that stage for a long period of time before you start over again. So in prophase 2, your nuclear membrane breaks down, your centrioles are going to move apart. All those things that happen in prophase 1 of meiosis and prophase of mitosis are going to happen again. In metaphase 2, this looks a lot more like mitosis. The chromosomes are going to line up individually along that metaphase plate in the middle of the cell. Just now we have half the amount of chromosomes for haploid. And we're again going to skip drawing the anaphase 2 stage, but remember in anaphase, that's when in this case, our sister chromatids are going to separate. So the sister chromatids on this half of the plate are going to migrate to this end. The sister chromatids on that half of the plate are going to migrate to that end. Same thing in the second cell. And so in telophase 2, then we're going to end up with our final gametes. And so because of crossing over, each one of those new cells is going to have a slightly different combination of genes from mom and dad. From their grandmothers and grandfathers. Yeah, sorry, I know it's really low down. So, once you finish on that, I just want to give you some time to walk back through that whole process, either on your own or with your group members. So just take some time to walk back through your diagrams and see if you can kind of, preferably with your shoulder buddy, because verbalizing it and saying it out loud, I think helps you to make sure you understand what's going on.
You can just get, yeah.
We're starting in paper.
This is Julia.
I have a question about the date, like signing up for IU. Do we have to do that again?
No, they only sent that out because some APC courses like speech are only in the second semester.
Oh,
So
okay.
as long as you are already registered, you don't have to register. Yeah, I don't know why they can have that up.
I've gotten like 8 of them in the past week.
Really?
So we
I'm
have to,
sorry. You don't have to register twice. If you already registered once, you don't have to. That's just for the classes that are only second. I was gonna do more lecturing today, but I think we'll stop and cover the rest of it tomorrow.
So we're not taking a quick break tonight?
No, the quick check